So now it's my great privilege and personal joy to introduce our keynote speaker, Professor Chng Wee Joo, the Yong Loo Lin Professor in Medical Oncology at National University of Singapore. So I've had the privilege and honor of working with Professor Chng for over a decade, and I can certainly say, not just for myself, but also I think for many of us who work with him very closely, that his mentorship has been invaluable to us in shaping our professional journeys, especially in instilling in us a resilience to overcome obstacles, as you know, that often come our way when we do our research, as well as the courage to push the boundaries of our work. Right? So, in addition to his professorship, Prof. Chng also holds several key leadership roles, including the Vice President of Biomedical Sciences Research at NUS, Group Director of Research at the National University Health System, Executive Director of the Singapore Translational Cancer Consortium. Now, he also chairs the Asian Myeloma Network and serves on prominent international professional bodies such as American Society for Hematology and the International Myeloma Working Group. As a renowned hematologist and leader in cancer research, he specializes in multiple myeloma and other blood cancers with pioneering work in genomics and therapeutics that has significantly improved patient outcomes worldwide. His contributions have in fact been recognized with numerous accolades, including the International Myeloma Foundation, Brian G. M. Dury Outstanding Achievement Award, making him the first in Asia to receive this honor. So this evening, Prof. Chng will deliver his lecture titled Curing the Incurable, the Past, Present, and Future of Multiple Myeloma, sharing the remarkable progress in myeloma treatment over the past 2 decades, the lessons learned along the way, and the exciting possibilities that lie ahead as we work towards curing a greater proportion of patients. So please join me in welcoming Professor Chng Wee Joo.
Thanks, Huda, very much for the very kind introduction and a warm welcome to all. So nice to see so many friendly faces in the audience. Special thanks again to have in our presence the Chairman, NUS Board of Trustee, Mr. Xie Fu Hua, Ms. Yong Ying Yi, the Dean, School of Medicine, Chong Yup Sing, CE, NUHS, Professor Yeo Kei Guan, and my boss, Professor Liu Bin, who is the NUS DPRT. Before I start my lecture, I just want to say a word of thanks to Professor Yoshi Ito, who before me for many years was the Yong Loo Lin Professor in Medical Oncology until his retirement in 2022. What is unknown to most is that Prof. Ito gave me my start in research, because before I went to the Mayo Clinic for my research fellowship, I have never worked in the lab before. And he was kind enough to take me on for 6 months working in his lab in IMCB, in the Biopolis, taught me a lot of things at the beginning about research. And it was a privilege years later for my group and his group to be able to work together with Siok Bien, who's here in the audience, on a project looking at one of his favorite gene, RUNX3, in lymphoma. So it almost felt that things have come a full circle for me to deliver this lecture today. So we're going to talk a lot about myeloma today. So what is multiple myeloma? It is actually the second most common blood cancer in adult. And it arises from these cells called plasma cells, which are normal immune cells that live in our bone marrow. They are important for immunity because they produce the antibody that help us fight infection. But in myeloma, what happened is some of these cells went rogue by acquiring cancer-causing gene mutation and can expand in the bone marrow. And these myeloma cells produces monoclonal protein that can be easily measured in the blood. So this is a big advantage for myeloma because we have an easy blood-based marker to actually make diagnosis and track disease progression. How do they cause health problem? What happens is that these myeloma cells secrete factors that erode the bone, cause holes in the bone, or what we call lytic lesions that may cause bone pain and sometimes fracture in the bone as well, which can be quite debilitating. The monoclonal proteins that they produce are excreted in the kidneys, and therefore, when high amount of these proteins are excreted, may cause damage to the kidney. And when the plasma cells that are malignant begin to grow and expand in the bone marrow, They may crowd out the normal blood-producing cells, causing low blood count and all the consequences. And they may also alter calcium metabolism that lead to high calcium level in the patients as well. But before patients become symptomatic, there is always this pre-malignant asymptomatic phase called MGUS, or monoclonal gammopathy of undetermined significance, where the clone is very small and they produce small amount of these monoclonal protein. In fact, this condition, MGUS, is very common in older people. Those above 60, there's about 10% that when you check their blood, you'll find these monoclonal proteins, but the individual is completely asymptomatic, and they will progress to myeloma only at 1% per year, which means that most people with MGUS do not develop myeloma in their lifetime. And then at the other extreme, at the advanced stage, the myeloma cells that normally live in the bone marrow and need the bone marrow for the microenvironment for its survival can actually develop ability to escape from the bone marrow, circulate in the blood, and form tumors outside of the bone marrow, what we call the extramedullary myeloma. What is most exciting about myeloma is really in the area of drug development. So for a relatively rare cancer, myeloma drug development, I think, beats every other cancer around the world. 14 new drugs approved since year 2000, 9 since 2020, and these drugs are better and better compared to previous ones, and approval is, and drug development is accelerating. And this has led to progressive improvement in the survival of myeloma patients over the years. Although today it's still generally incurable, because what happens is that patients have effective treatment, go into remission, but the disease tend to come back some years later, and then you need to give them a different type of treatment again, which means that we can use drug to control myeloma, but not to cure it, right? So in a way, myeloma is becoming like a chronic disease in the sense that like diabetes or hypertension, where you need to take drug regularly. But still, despite the improvement in outcome, we see that 25% or a quarter of patients may live for less than 3 years. So these are what are the high-risk patients. So the current state of art in 2025 is that there is very rapid drug development because of increasingly better understanding of disease biology. We can stratify patients. The drugs we have are many and very effective. And the good thing is that they're given outpatient. Patients do not need to stay in hospital, right? And the side effects profile are very positive. So patients also have good quality of life in general, although they need to take medicine continuously. The average survival now is 8 to 10 years. And on the right, you see data from our center in NUH, where the outcome is 9.1 years, which is right there in the international benchmark. And about 15% of patients have long remission and may well die of other conditions, not myeloma, and they potentially have cure from the condition. But looking forward, it's even going to be more exciting because this latest trial using a combination of 4 drugs in younger patients that are fit for stem cell transplantation is showing that they can have remission for up to 17 years before they have their first progression. Compare that to the 9 years in the previous slide is a great leap forward. But it's going to be even better going forward. Why? Because we have better, newer drugs in the horizon. So this slide is showing that today, if you have a patient at 3rd relapse or more, and you give them a treatment, they are likely to only have remission for 4.6 months. Okay, remember that number. And they're likely to not survive beyond 12 months, 1 year. But in this same type of patients, the latest trials using CAR-T targeting BCMA, which is a protein expressed on myeloma, is showing that 1/3 of these kind of patients has progression only after 5 years. And compare that to the 4.6 months, again, is a fantastic leap forward. And if we can bring this effective treatment earlier on in our treatment lines, potentially the effect will be even better. So the prediction for myeloma patients going forward in 10, 15 years' time is that we potentially could cure up to 50% of patients with a survival of more than 20 years. And there will be increasing use of CAR-T in earlier lines of treatment, potentially even replacing stem cell transplant in the upfront setting. And the great advantage of CAR-T is that it's a one-off treatment. You don't give people repeated doses of CAR-T, and that will mean that we are achieving all this good outcome with more limited duration of treatment rather than continuous treatment. So really, myeloma patients have a lot of positive things to look forward to. But that's not always been the case. When I started research in myeloma 2 decades ago, the median survival is only 3 to 4 years. There are very limited treatment options, all of which are chemotherapy-based. Patients have to do that in the hospital. Stem cell transplant is the mainstay of treatment. And at that time, some of the novel agents are starting to just emerge. And we know very little about myeloma biology, and it's just starting to use genomic techniques to understand myeloma. So I've been pondering this question of how did we get here? How did a rare type of cancer like myeloma become such a mighty model for drug development and improvement in outcomes for cancer patients? And I think these are the key elements. And I think in many diseases, we see some of these elements, but I think in myeloma, we see all of them coming together in a very, very effective manner. So one is this understanding of biology through the study of genomics and, of course, other new areas of science to drive the clinical development of treatment. The research agenda is often determined by clinician scientists. And I think this is key, and this gives me the validation of why Singapore should be pushing for more clinician scientists who are passionate about solving problems for their patient. And because clinician scientists are not biased towards any specific technology or area of science, we often try to find the best solutions for the problems that we're seeing in our patient. And like our dean liked to say, be married to the problem, not the solution. And so, we are agnostic about what solution as long as it works and help our patient. And amongst these clinician scientists are also strong leaders that are good at organizing the team, growing the team, and who also recognize for a rare condition the importance of collaboration. So these leaders collaborate across regions and across the world to find the best solutions for the patient. So, again, following this edict of if you want to go far, go together, I think we best exemplify this in the myeloma research community. In addition, the leaders are able to convince donors and philanthropic organizations to buy into the research agenda. And I think this is critical because often donors donate for education to help patients get access. But if you don't drive the research agenda, the progress will not be seen at this kind of rates. So we have International Myeloma Foundation and the Myeloma Research Foundation that are very much involved in generating a lot of research data. And when these data are generated, they are openly shared in a transparent way so that more people can come in and work in this area and to accelerate progress in research. And in terms of drug development, it is important to work closely with industry that produces the drug to accelerate progress. And this partnership is very strong in myeloma. So on this slide, you see all the big names in myeloma. For those people in medicine, you may recognize some of the name. They are there, brave enough to ask the big question and to do needle shifting kind of research. And I just want to highlight Brian Dury, who is the scientific chair of the International Myeloma Foundation. He was instrumental in bringing experts from across the globe to come together to form the International Myeloma Working Group. But not just that, he funded through the International Myeloma Foundation this sort of ambitious, moving the needle type of research towards cure for myeloma that he called the Black Swan initiatives, because these are Black Swan events, supposed to be rare events, but he wanted to be able to facilitate the development of rare events for myeloma patients. And the International Myeloma Working Group has now grown to a size exceeding several hundreds experts across the globe, including from Asia, Latin America, Europe, US, Australia, and so on. And this is the group that produces all the guidelines to help people practice properly and treat myeloma patients as best as they can, but also come up with criteria, definitions to standardize inclusions of patients into trials and reporting of trial outcomes so that between the different trials, we have parameters that we can trust and can be comparable. And I have been very privileged to be part of this group for slightly more than a decade. And we have a meeting every year in a different European city called the IMWG Summit. So this is a picture taken from one in 2024. But this is a very special meeting. We all know about cancer conferences and so on, where People go and learn because it's educational in nature, or to share their latest research for exchange. But this meeting is just with the expert that look at what are the current gaps we have in this disease to formulate the next stage of research agenda. And in the same meeting are potential donors, are industry, to work with us towards achieving those goals. So the agenda is determined by the clinician scientists, by the experts, not biased towards any specific company, technology, biomarker industry, and so on and so forth. And I think that's quite unique, actually, and something that perhaps we should think about more. And because our outcomes are getting so much better, like I said, the survival is going to many years. If we approve drug using the conventional endpoints of survival, we're going to take many years for new drugs to get approved. We have to do trials that last for more than 10 years, very costly, and it takes a long time for drug approval. So the International Myeloma Working Group actually developed a minimal residual disease test which can detect very low level of disease very sensitively, and also collected a lot of data over the world to show that this minimal residual disease test is a very good surrogate endpoint for survival. When you have improvement in MRD rates, your survival will improve. And they went with this data to the FDA and got a unanimous vote approving the use of MRD testing as an early endpoint for accelerated approval of myeloma drugs in clinical trials. This endpoint is at 12 months. So now, despite our survival getting longer and longer, we can still get new drugs approved after 12 drugs using this surrogate endpoint. And that just strikes me as a very powerful message, because clearly international collaboration, if done right, can actually move the needle even on policies and regulatory requirement for approval. And I've taken many of these insights and lessons towards our own team, building our own team in Singapore and across the region. So today I'm very happy to say that we have a very strong team working on myeloma in NUHS. These are all clinician scientists. Melissa, Sanjay, Sini, and even our advanced practice nurse, Chng, are all interested in research. We either all have or undergoing training with a PhD. And I would say that this in Singapore is very rare. In fact, probably the only kind of disease area where everyone is a clinician scientist and doing a PhD. We have established many clinical resources, patient database, myeloma cell bank, and so on, that we are very open to sharing with the research community to come in and work with us in myeloma. And I think you'll see in some of the subsequent slides that collaboration is a big theme of our research program. And we have very good collaboration across the whole of Singapore in the Singapore Myeloma Study Group with our colleagues in SingHealth, NHG, as well as in private sector. And this is a strong foundation for us to also do a lot of good work in Asia through the Asian Myeloma Network, which was established in 2012, again under the leadership of Brian Dury and the International Myeloma Foundation, because they recognize that there is a lot of myeloma patients in Asia, because we have a lot of people living in Asia, but hardly anything is known about Asian myeloma. Is this exactly the same as in the Western? Is there some unique features? And so this was established, and I was one of the founding members, and I'm glad to say that today I'm chairing this network. We have now grown to more than 200 members from more than 60 sites across 11 countries and regions in Asia. One of the early tasks that I was given in this network is to establish a clinical trial network for doing trials across the regions involving multi-centers and multi-countries. This was clearly a very challenging task because every country has different regulatory requirement and ability to do trials. But we have succeeded in establishing a very sustainable mechanism for doing trials for more than a decade. We felt that this was an important thing to do for Asia because we have clear data that patients with early access to novel drugs through clinical trials do much better than those that do not. So this is important for our patients, and trial is a good way to get early access to drugs. So we've done many trials. A lot of them has completed, published. Some of these results have been used by certain regulatory jurisdiction, like in Taiwan and so on, for labeling or approval of drug combination. These trials have benefited more than 400 patients across Asia, the mechanism is still working very well, and we have more and more new ideas that are coming online. And we're extending the network. We have now got consistent collaboration with our Australian partners. We're building new bridges to Latin America, to Europe, as well as to Canada and the US. And we're now embarking on our next stage, which is a very ambitious Asian myeloma multi-omics project, or what we call AMO, because we are building the bullets to kill myeloma. And we are collecting samples from across the regions to do multi-omics genomic studies to answer some important questions in myeloma. Why is the incidence of myeloma lower in Asia? It's almost half compared to the West. Why? Is there something protective in our germline genome that is preventing this? So this is something that we want to find an answer to. We're also looking at difficult to treat diseases that are still left, the high-risk patients, patients that progress early, that are refractory to very good treatment that we have today, or patients that develop extramedullary disease. And we want to find new treatment that is possible for these kind of patients. And the data generated, we are collecting into a database that we call the Asian Myeloma Atlas, that again, we want to make freely available to the international research community as our contribution back to the ecosystem. So what has been our scientific contributions? And it has always been an area of genomics. I've been working on myeloma genomics for 20 years, and we are using this as a way to understand biology in this complicated disease. So when I started, it is very clear that there is two patterns of genetic changes in myeloma. One are these translocations, or when two pieces of chromosome come together to form a fusion gene. We can easily detect this using a technique called FISH, or fluorescent in situ hybridization, which is seen on this picture. You can use this fluorescent probe to detect the joining of two different pieces of DNA. And this is very valuable for myeloma because myeloma cells are not very proliferative. They don't grow very fast. And this technique allows us to detect genetic abnormalities in non-dividing cells. And so we can apply it to all myeloma patients. The other pattern that is observed in those days was this hyperdiploidy, where they have trisomies of, which is 3 copies of a number of chromosomes. You can see on this picture, 3 copies of this chromosome, 3 copies of this chromosome. And this requires a technique called karyotyping for us to see these different chromosomes, and that can only be done on tumors that are proliferative, that are growing fast, which is only a small fraction of myeloma. So we have a conundrum because we can look at using FISH in the earlier stage of the disease, which is MGUS, and we detect these same fusions in MGUS as compared to the myeloma. We know that the fusions exist from start to diagnosis, right? But we don't know whether the trisomies also come from the cells with the fusions through evolution and changes, or they come from a completely distinct set of cells. And so what we did in those days was to develop a FISH-based assay to detect trisomies and hyperdiploidy. Apply this to MGUS, and what we see is exactly the same pattern of change of trisomies that we see in myeloma is also seen in MGUS, and in fact, this pattern is completely mutually exclusive to those with a translocation, or at least in 90+% of patients, it is completely exclusive. So defining two distinct genetic evolution pathway for myeloma. But like I said, it is already there in MGUS, it is also there in myeloma, and I said earlier that only very few MGUS will develop myeloma. So surely there must be a second hit, a second event that lead to this progression. And this was what we then use whole genome microarray technique, because we're fishing for something that we don't yet know about in the entire genome, to try to pick out the signals that will guide us. And what we saw was overexpression of a set of genes you can see in red here, which is high expression, in myeloma, but not seen in MGUS. You can see here is all blue. And what these gene signature tell us is that there is an activation of a gene called MYC, which is a very well-known cancer-causing gene. And working with Seok Bien, we then identified that this is indeed the case specifically in the plasma cells of myeloma, where MYC is activated, but not in MGUS. So we found that there is unique second hit or secondary pathway that is distinct between the different genetic subtypes of myeloma over the years. And working from that, we further define really a lot of different genetic subtypes in myeloma, the important genes that the genetic abnormalities are driving, the biology that is underlying what these genes are doing. We then went on to the clinical samples to define their frequency. How does this genetic aberrations affect response to drug? And also, what are the outcomes of the patients? And synthesizing this body of work, we can clearly see that there are high-risk patients that live for a very short period of time, but there are also good-risk patients that live for a very long period of time. And I was tasked by the International Myeloma Working Group about a decade ago to introduce this concept of risk stratification in myeloma. And this has become today very important because we see that clearly, despite all the improvement in our treatment, the high-risk patients are still doing very badly. And this sets the framework for us to design clinical trials specifically for high-risk patients to see whether we can find different therapeutic strategies to improve their outcome. And in fact, the current problem that we're trying to solve, one of which are really how to improve the outcome of high-risk myeloma, which today is a real unmet medical need. And similarly, we're taking, for us, a genomic approach because there are genetic aberrations that are closely associated with poor outcome, one of which is this 4-14 translocation, which is a fragment of chromosome 4 and 14 coming together, resulting in very high expression of this gene called NSD2, which is an epigenetic enzyme that modulates the expression of a large number of genes. So NSD2 is not targetable. There's no drug that targets NSD2. So our hypothesis is that if it leads to modulation of expression of large number of genes, maybe some of these genes are actually membrane protein that is expressed highly in the 414 myeloma, but not other cells or other myeloma. And we can develop an antibody that actually can target this protein to eradicate the tumor cells. It turns out there are some of these highly expressed proteins, but they also have some expression in other normal cells, which means that if we just use the antibody, it may have some side effects. So we then have this idea that if we use the antibody to deliver a second bullet that is specific to the tumor because they are dependent on certain genes for their survival, then we can have the additional specificity for this tumor. So this is a project that we're working together with Prof. Fan from NTU and Kong Ping from the Immunology Network to develop what we call antibody nucleic acid therapeutics, where we can use antibody to deliver specific RNA to knock down important genes in cancer. And our early molecules have been very successful in knocking down the gene and protein of interest and also killing the myeloma cells in cell line models. But more excitingly is also that it has the same effect in patient samples as well, showing that the potential for clinical translation is there. The second group that we're dealing with are those with a 14-16 translocation that overexpress a transcription factor called MAF. Transcription factor regulates gene expression as well, but it is not easy to target. There's no drug that targets MAF today. So again, we have to use a different approach, is to understand what is the biology that is unique for these cells that have MAF overexpression. And in our work done in collaboration with Wai Leong, who is one of the key cancer metabolism expert in GIS and CSI, we found that MAF deregulation is associated with a unique metabolic shift so that the cells are now addicted to glutamine, which is a nutrient that is present in the microenvironment. And what happens is that MAF drive the expression of glutamine importer so that there's a lot of import of glutamine into the cells, and over time, they become addicted to this. So if we block the import of glutamine or deprive glutamine from the environment, the cells most likely will die. And in fact, this is what we observe in our experiments. And this seems to be very unique to the MAF tumor because other kind of genetic types of myeloma do not seem to have the same effect. So we're working with Wai Leong and others to see how we can translate this finding now into something that we can apply to patients, either to dietary modification or specific drugs that block the import of glutamine in the cancer cells. The last high-risk group are those with 1q21 gain. So this is chromosome 1, the long arm, and extra copies of this long arm. And working with Polly Chen at NUS School of Medicine as well as the CSI, we identified that ADA1, which is an RNA editing gene, it changes RNA sequence, is a key oncogene on 1q21. It is very highly expressed in patients with 1q21 gain, but not just that, it is functional, because we find that the myeloma transcriptome is hyper-edited in these patients as well. And we have shown that ADA1 is oncogenic, right? It leads to increased cell growth, increased formation of colonies, increased tumor size in animal, and when we knock it down or inhibit its function, the tumor reduce, the cell growth reduce, and the colony size reduce. So now we're working with Polly and others to actually identify potential ADA1 inhibitors that we can try to develop towards the clinic. And the last thing I want to talk about that we're working on is really this whole idea of precision medicine. And I will tell you why myeloma is a perfect model for doing this kind of work. Because, as I said earlier, we have so many drugs that is effective and approved. When you look at combinations, the number of potential combinations are many, many. When we have a patient that relapse sitting in front of us, what is the best treatment to choose for this patient is a real conundrum, right? And it will be, so the concept of personalized medicine really is very relevant to the treatment of myeloma. So we've been working with Ed Chow and Dean Ho from NUS as well, and Ed has now gone back to the US to run or work on his company, Cayenne Technologies, and we have people from Cayenne here as well, on co-developing this QPOP platform for myeloma. This is a AI-based modeling platform that is able to find the best drug combination out of a fixed drug set of drugs, using the minimal amount of experimental data that is required. And in fact, one of the earliest studies using this QPOP platform was in myeloma, where we found and identified novel combinations that have never been tested in patients that work in myeloma. But that work was in a cell line, and when we tested it in patients, it doesn't seem to predict as well. And I think we know the issue is that, as I said earlier, myeloma cells is very dependent on its microenvironment in the bone marrow. And when we try to apply this technique in the Petri dish, we are not replicating the bone marrow microenvironment. And so in fact, this is one of the critical pieces that we're working on together. Sanjay and Liren is working on this piece to define what are the critical factors that is needed to replicate the human bone marrow so that we can culture the cells in this better system to get better fidelity from the prediction. And not just selecting the right combination. I think we can do even one step better, which is the best doses of these drugs to use in individual patient. And that Sanjay is now leading a trial together with Dean's team using his Curate AI to modulate drug dosing in patient. And why is myeloma ideal for this work? Because we have a blood-based dynamic marker that we can measure regularly to check on response. It's not easy for other tumor where you have to take repeated scans or biopsies, which are not easy. So I think I always felt that myeloma is an ideal model for this. And lastly, I told you that patients that are high risk will likely have residual cells that are resistant to treatment. And so we want to have an ability to collect these residual cells to understand why they're resistant to treatment. So we're working with Tritik, I'm not sure whether he is here today, on developing a microfluidic platform that can harness malignant circulating plasma cells so that we can subject them to single-cell genomic sequencing and understand why they're resistant to treatment, and then iterate this process again to find the best combinations for these rare resistant cells to further help to eradicate patients with high-risk disease. So I think in conclusion, hopefully I have helped you understand that myeloma has sort of really improved tremendously in terms of the research, the drug development that has impacted our patients a lot. I think there is good reasons why for rare conditions we have been successful, and I hope that I share some of the insights which we hold very dearly to our heart and try to replicate in our own research group. But there remains a high-risk group of patients that still have very poor outcome. And I think the approach to this is to better understand the biology of high-risk patients, to better enable the next phase of drug development, And myeloma is very heterogeneous. Even when we talk about high risk, it's not just one high risk, there's many different types of high risk. And we need to take a more biological, knowledge-based, targeted and personalized approach to eradicating myeloma from these patients. So I just want to acknowledge many people. I won't go through them, but we really have benefited from so many collaborators from across NUS, NTU, A-STAR, and also overseas that have brought their knowledge, expertise into the myeloma field that so much enriched our research ecosystem. All my clinical collaborators locally and overseas, our own clinical teams, and of course my own research lab that has put in so much effort working on myeloma issues over the last 2 decades. This is just to show that we don't just work, we also know how to play hard. This is our annual Christmas party. And last of all is really the patients who has continued to inspire and motivate our work. And we are, after all, doing all this research to help them improve their outcome. And just to say that they often contribute back to us by raising funds for our research as well. So just want to thank them from the bottom of my heart. And with that, thank you very much and look forward to some questions. Thank you. So anyone with questions, you can just raise your hand. We have colleagues at the side that will be happy to pass you the mic. Yes, sir. Mr. Ramesh.
That was an outstanding overview, and thank you for sharing that. You mentioned early on about CAR T, and the question I have is, what are some of the scientific and non-scientific barriers to using CAR T as first line in multiple myeloma?
Yeah, so very good question. I think as with most drug development, we need to first prove that something is effective first. And usually in cancer, we do that in the late lines of disease because there are no good alternative, right? So clearly that was what is done here, and I showed some outcomes of that, and the results are very good. So in fact, the clinical trials are now people are doing now incorporate CAR T into the front line of treatment. So we really need to prove that in fact is better than the current treatment, which I show is producing potentially 17 years of PFS, right? And the danger with CAR T is that they may have some of their toxicity as well. The experience with CAR T is still relatively short. The danger with some of these novel agents is that they may have long-term side effects which are maybe unpredicted for or unknown yet. So I think to move something like that up front, which is very exciting, obviously, we have to be cautious as well because the current standard is potentially giving patients a long period of progression-free survival. So you don't want to jeopardize that willy-nilly as well. Yeah. Yes, maybe Yunsin first, and then we get, maybe Ashok first. The mic is nearer, but we have Dr. Chen here as well. Yes, Ashok.
Wee Joo, thanks for the lecture. You mentioned early on that we don't know whether Asian myeloma is distinct from myeloma elsewhere. And this is a problem that I think we encounter in Singapore over and over again. Is Asian disease biology really different from disease biology overseas? So one question I had, and it's particularly perhaps answerable in the context of a well-characterized disease like myeloma, is this. We now have people of Asian genetic descent who have spent 2, 3, 4 generations in Western countries. If it's genetics and not environment, they too should have less myeloma than people of European descent. Do we know this?
Yeah, so we don't yet know, but we know that there are some racial differences because myeloma is much more common in African Americans versus Caucasian. And really across Asia, it's the lowest incidence. But we don't yet have data. For example, I think what you're saying is that the Asians that have lived for 2 decades in America, whether they are also lower or not. And the problem of that is that often Asians are excluded in studies, that is an effort that needs to be organized.
It's not 2 decades, really. It's several generations. There are people of Asian descent who have been born and brought up in the
but they
West,
are generally underrepresented in a lot of the studies. So I think it needs to be a deliberate approach there. And so one of the reasons why we wanted to do the germline study as well is to see whether indeed we see some unique signals from here, and that will dovetail very nicely to our own precise precision medicine program, which is normal germline, and we have myeloma germline. And there are many of these large-scale germline consortium of European and US that we can also make comparison with. So I think that would be useful. But what is clear is that we do see differences in response to drugs, as well as sometimes side effects in Asian compared to the West. Dr. Chen, yeah, Vincent? Yep.
Thanks, Wee Joo, for your very inspiring talk. Always like a privilege to work alongside with you as well. I think my question is that the developments of this biotech has been very rapid in the last few years, and we have many agents that can actually benefit patients. But the developmental timelines are all getting shorter and shorter. And with this such a landscape, how do you foresee that we can try and quickly clinically translate these to our patients such that they benefit, and how maybe Singapore as a whole can select some of these best treatments outcomes that will actually make a difference to our patients?
Yeah. So very good question. I think the approval and development is very fast, but actually when I talk about approval, it is US, European, mainly US-based approval. So when we talk about Singapore, there's a lapse, there's a lag time. Not all the drugs that are here are freely, easily available here in Singapore. So I think there is a challenge. There is a challenge there, and I don't have an easy answer to that, except that we need to continue to maybe work with our regulators and MOH to see how best to help patients. We have to also potentially think of two things. One is that I think it's coming to a stage where indeed some large percentage of myeloma patients can have very, very long remission or cure with just current standard of treatment. So we need to make sure that they get hold of that. And number two is that really explore the possibility of using things like QPOP AI to predict the best drug for individual patients so that we can also achieve the best outcome for them. Because patients can survive a long time even with just current treatment, actually. So we shouldn't forget about that, but it's the relapse, the high-risk patients that then run into issue, and we want to have as much available for them as possible. And today we have quite a lot, actually, but we are always behind the curve, like in that sense. I agree with you. That is a challenge that we have to work together with regulators on as well. It's not just us. Yes, Vinay?
Thanks, Wee Joo. I was wondering about the conversion of a normal cell to an MGUS. You talked nicely about MGUS converting by 1 or 2 hits into cancer cell. Is there anything known about clonal hematopoiesis, which also is seen in a large number of patients, as is MGUS? And are there any common mechanisms? What would you predict?
Yeah, fantastic question. Today we were just talking, I think Anand was on the call, we were just talking to a potential recruit about clonal hematopoiesis because he's interested in that in the leukemia field, right? But indeed, clonal hematopoiesis have been identified in patients with myeloma, but mostly the studies have looked at, what is the risk of, say, secondary malignancy with transplant from that kind of perspective. But I was thinking that perhaps there's interaction between these clonal hematopoietic cells, the microenvironment, and the development of myeloma. What is clear is that the microenvironment and the immune system is very important in the progression from MGUS to myeloma, actually. But how exactly is it doing is currently unclear, right? People are studying that. But the establishment of the myeloma, the MGUS cells, I suspect is just an error, because myeloma is generally a disease of the elderly, and there's ample data in cancer that as you grow older, because the error in your DNA repair, chances get higher, then one day you get this unlucky, you hit one bad gene in a bad cell, and then you establish the clone, right? I suspect myeloma, the initial establishment of the MGUS clone may be somewhat related to that, because people have studied the progression from MGUS to myeloma, but not really on normal cells to myeloma, like how did that happen? Yeah.